humanity is struck and the whole world has gone into lockdown. Stay tuned for this episode where we'll be unraveling the mysteries of the horseshoe crab and its blood, which is the key to unlocking the COVID-19 vaccine. Welcome, students and listeners. My name is Alice Furman. I'm currently a biology major at BMCC, and I'm joined here today by my two fellow experts, Audrey and Omar.
Hello, everyone. Thanks for joining us. I'm Audrey Hedges. I'm studying biotechnology at BMCC currently. I'm so excited to get started.
Hi, guys. I'm Omar, and I'm currently studying to be a veterinarian at BMCC with a biology major. So I had a question. What exactly is a horseshoe crab, Audrey?
Good question, Omar. A horseshoe crab, also known as the Limulus polyphemus, is what I like to call them the tactile spider, is a living fossil walking among us. They lived for over 450 million years, predating the dinosaurs. Funny thing, they aren't actually related to crabs at all and are more closely related to the spiders and scorpions. These cute little guys, if you ever get the chance to see them, look a lot like an upside down bowl with 10 creepy legs on the ventral half and of the body and a telson extending from the caudal end of the body. Some might think the telson is a stinger, but it's actually for digging and to help the horseshoe crab flip right side up if it ever happens to flip over. So basically, they're harmless, creepy little spider scorpions.
Wow, that's amazing. Tactical spiders, you say? They're like the tanks of spiders? With that note, let's just buckle up and jump into our time machine and back to the year 1956, where Dr. Frederick Bang discovered the horseshoe crab's secret to unlocking our modern-day vaccines.
So the year is 1956. Dr. Frederick Bang is taking a lovely stroll on the beach when he sees something amuck in the sand.
Dr. Frederick Bang was a doctor who applied marine biology to modern-day medicine, and when he discovered the blood clotting in the horseshoe crab, he had to know why. Eventually, he came in contact with a well-known hematologist at Hopkins University, Dr. Jack Levin, and together they developed the Limulus lysate test after discovering the blood clotting agent, Limulus amoebocyte lysate, in the horseshoe crab. Because the horseshoe crab has an open circulatory system meaning that the heart pumps the blood to the blood vessels, which then enters the body cavities to the organs. They're all bathed in blood, unlike humans who are a closed circulatory system. So we're not sitting around in our own juices. So this has been working great for the horseshoe crab for 500 million years, so no complaints there. So let's go back to the future and learn more about this amazing defense system.
We've entered the present day. The horseshoe crab's blood is at work again, saving humanity. As good as horseshoe crabs are at detecting endotoxins and bad bacteria and the things that go in our body, it also has a really neat immune defense system. Different from the systems we learn about in class, the horseshoe crab doesn't have a typical immune system. Instead, it has lots of defense molecules and components that together create the horseshoe crab's innate immunity. In chapter 43 of the immune system, we learned that innate immunity is present before any exposure to the pathogens and is effective from the time of birth, responding to a broad range of pathogens. And you can find this in all animals and plants. This helps the horseshoe crab protect themselves against all sorts of pathogens, all thanks to a type of cell in its blood, an amoebocyte. This allows them to detect endotoxins, which are chemicals found on the outer membrane of certain species of gram-negative bacteria. When amoebocytes get in contact with endotoxins, they release a clotting factor known as coagulagen. The coagulagen activates defense molecules that neutralize the pathogen. Once it begins to coagulate, it forms a mass around the pathogen and confines the bacteria to one area, preventing it from spreading to other parts of the crab.
Fun fact, and this actually references back to our class discussion with Dr. Joanna Giza about crayfish. They're similar to the horseshoe crab because the blood contains hemocyanin, right? So it is a copper-based protein that turns blue when oxygenated instead of the iron-based hemoglobin in red blood cells found in vertebrates. As an additional similarity between the crayfish and the horseshoe crab, the horseshoe crab's blood also has a chemical called coagulin that can detect bacterial contamination at extremely low concentrations. There are many labs that are using this very technique, and one of the main horseshoe crab labs in the United States is Charles River Laboratories. It's based in Massachusetts. Horseshoe crab blood is collected by Charles River for biomedical research and for the production of limulus amoebocyte lysate. Alice, I know you did some great research on this topic. Can you tell us a little bit more about this?
Yeah, so there are actually about 5 to 6 LAL laboratories in the country. But surprisingly, although it's such a big contribution to biotechnology, LAL harvesting is done under top secret conditions that are not shared with the public. In America, the FMP, or the Interstate Fishery Management Board, which establishes the biomedical mortality threshold. So as you mentioned prior about the Charles River Laboratory, one of the leading LAL harvesting centers, they say that they stress the importance of having low mortality against horseshoe crabs, with this facility having a mortality rate of only 4%. At Charles River, they do necessary endotoxin tests, and this works by first taking blood from horseshoe crabs. They only take about 1/3 of their blood at a time, and this can usually take about 2 males to fill up a specimen bottle. But a female, which is about 25% larger than the male, can fill up a specimen bottle by themselves. So once their blood is taken, they're then returned back to the water. So back at the lab, the white cells that contain the special protein settle to the bottom of the processed sample. Once the blue plasma is skimmed off, the remaining material is freeze-dried into the powder that they use in biomedical testing, ensuring that they really refine it well. And it leaves them with this LAL reagent that is the standard testing to ensure anything that may get in contact with blood, as well as making sure all of our vaccines are free of endotoxins.
That is so interesting. It really shows why they've survived as long as they have. Thanks to the horseshoe crab, there are many different vaccines in the making currently. The one that is in the forefront of the race to create COVID-19 vaccine and is being made as we speak is a vaccine by Pfizer and BioNTech, which, fun fact, was developed by a Turkish-German power couple by the name of Uğur Şahin and Özlem Türeci. They are both trailblazers in the field of oncology and infectious diseases. The vaccine has been proven to be 90% effective in preventing infection. This vaccine uses RNA messengers to help the immune system fight off infection, which is something we learned about in class. The RNA, or sometimes called the mRNA, is vital for the body to communicate. There are messengers of all living cells carrying instructions from the DNA for controlling the synthesis of proteins, changing the expressions of the genes, and therefore altering the DNA itself. What Şahin and Türeci essentially did was train the immune system to build up more antibodies by having the mRNA produce the proteins itself. The protein being the spike protein, which is harmless on its own, but is the key to triggering the immune system's defenses, allowing the body to create a surplus of antibodies to defend itself from the virus. This is the fastest vaccine to ever be produced in history.
That is incredible, Audrey. I cannot wait until it's open to the general public and that we can all get back to our regular lives. All of this wouldn't be possible if it wasn't for the LAL, which is used in bacterial endotoxin testing systems, as it is the most specific and sensitive method available for endotoxin detection. Is there anything you want to add to that, Alice? I knew this was one of your favorite topics.
Yeah, so did you guys actually know that there's a completely synthetic alternative of LAL that can be used for endotoxin testing? So it was actually discovered in 1997 when scientists at the National University of Singapore, Lin Ding Jin and Bo Hao, discovered that cloned laboratory synthesized recombinant factor C could be used towards an animal-free endotoxin detection technology. They began by cloning the DNA of a factor C molecule and synthesized RFC, the synthetic alternative to the LAL test. They then clone other factor C molecules and study them to develop various RFC tests. In the RFC test, the endotoxin activates with the synthetic RFC molecule, which cleaves a fluorogenic substrate and results in the generation of a fluorogenic compound. The fluorescence is measured 2 times, first at 0 and then another after the endotoxin has been introduced. The difference in the fluorescence is proportional to an endotoxin concentration and is what allows them to calculate a final endotoxin result. This is not something primarily adopted in biomedical research because there were concerns over the efficacy of RFC when compared to LAL. Many studies have been done on the efficacy of RFC, but overall results show that it is equivalent to the LAL test in its ability to measure and detect endotoxins. Funny enough, RFC factor does not contain some of the more sensitive factors that could subject the LAL test to a false positive. Hopefully in the future, we can work towards making RFC a more prominent endotoxin screener. But as of right now, there's only one synthetic LAL laboratory in the country.
Thank you so much for that information, Alice, and thank you, Audrey. That will be our ending to this podcast. Thank you to everyone who tuned in. I hope you learned something new today. And please remember your tactical friend, the horseshoe crab, the next time you get your vaccine, and hopefully it would be the COVID-19 vaccine. Thank you to the horseshoe crab for its contribution to the drug discovery. And if by any chance you see a horseshoe crab while you're taking a stroll on the beach on its back, do a good deed and flip them over. With that note, we're out of here. See you guys later.
Bye.